Michelle with Life Science Centre. There's been lots of talk about testing for COVID-19, and I thought you might like to find out a bit more about how these kinds of tests work. Now, we can't use any actual virus to do our testing for this video, so we're going to use different chemicals that simulate how these tests work. There are two main kinds of tests that are often used. Genetic tests start with a swab in your nose and your back of your throat and look for the genetic information from the virus. They can tell you whether you are infected. Antibody tests, on the other hand, will usually start by analysing your blood and can tell you if you have been infected and have since recovered. For the first test, to see if you have COVID-19, we need to see whether we can find any of the virus's genetic material in the sample. DNA is the instruction code for living things, and the information is stored in a double helix, two strands twisted together a bit like a ladder. Viruses, like the one that causes COVID-19, use RNA to store their genetic information. RNA is similar to DNA, but in this case only uses one strand instead of two. In any sample, there are only going to be a few bits of RNA, so we need to make lots of copies of it. First, the RNA is turned into DNA, which can then be copied millions of times by a machine like this one. Special chemical markers are added while the DNA is being copied. The markers bind to the DNA and glow under ultraviolet light. If the marker doesn't glow, there's no virus in the sample and the test result is negative. But the more virus there is in the sample, the more light the markers give off. Genetic tests for COVID-19 are very good for saying if you are infected, not so much for saying if you have since recovered. Antibody tests are much better for this. When your body detects an intruder like a virus, it starts to produce antibodies to help fight off the illness. Antibodies are perfect fit for a specific intruder, so looking for antibodies in the blood can tell us which illnesses your body has been trying to fight off. Antibody tests for COVID-19 typically look for two types of antibodies, IgG and IgM. Since they're produced at different times after the start of an infection, we can tell how long ago a patient was infected. In our simplified version, the test reagent produces a different colour to show which antibodies are present. Green means no antibodies are present. Blue means IgM. Yellow means IgG. Red means both IgM and IgG are present. Antibody tests are very useful for telling whether someone has had the infection and how long ago it was. However, having antibodies in your blood doesn't necessarily mean that you cannot be infected again. It depends how effective the antibodies are and how many your body produces. For some diseases, your antibodies can provide immunity and stop you from getting sick with that disease again, for either life, for years, or for just a few months. But COVID-19 is such a new disease, we just don't know how long any immunity might last. Remember, there are still lots of ways that you can help keep yourself and others safe. Wash your hands frequently with soap and water. Wear a face covering. Keep your distance from other people. And stay home if you have any symptoms or have been in contact with someone who had the virus.